Yeah.
I can't get my hair to do it. I like alfalfa in the back. Yeah.
No.
It looks like some are
I
coming
should
out.
have one of these guys in here.
So Chris is here.
Are you good to go?
We're getting started. It's just we've been having stuff, trying to get all the equipment to work. Not yet. Sorry. Oh, that's okay. I figured you guys were. Yeah, I bet. So I totally understand. So, do you know if anybody's
Oh, because it's him now. Huh? Change.
Of an idea of how he got to where he is right now. As you know, he's an associate professor of biology here at EOU, but a faculty member at the University of Washington Harbor Labs. He grew up in Palm Beach, Florida, and graduated with a BS in marine biology from Florida Atlantic University. He then received an MS in biology from the University of North Carolina at Wilmington and a PhD in neurobiology from the University of North Carolina at Chapel Hill. He then spent 5 years at Labs postdoctoral fellow, where he had his own independent lab, which received funding from NSF and DARPA. Is that how you say that? Okay. While there, he also guided undergraduate research apprenticeships. He also received a Woods Hole MBL fellowship to attend the Neural Systems and Behavior Course and Research Fellowship to conduct research at the Queen's University Belfast Marine Lab in Portaferry, Northern Ireland. For that, I'm jealous. With all of these amazing and what's likely a really busy schedule, we're really lucky to have him here today presenting some of his research. And please help me welcome Dr. Shaun Cain.
Well, thanks. I love doing these colloquiums. This is unfortunately only the second one I've done, but I think it's really important. It's good to see people coming out. We get to hear what each other are doing outside of the classroom and then developing our own research programs and so forth. So I think, and I appreciate you coming out, and I really love doing this. So what I'm going to talk about today is basically using a marine mollusk to understand the mechanisms that underlie behavior. And in this case, I'm really what's called a neuroethologist. So it's a component of neuroscience that looks at the nervous control of natural behavior. Most of the time in psychology, they're using very contrived environments to get at important questions underlying behavior. In this case, we try to look at how evolution has shaped nervous systems to allow organisms to respond appropriately within their environment. So that's what I'm going to talk about today, are a couple of projects that I've been working on with various colleagues and students trying to understand the nervous control of behavior. So I just want to give you an outline of the talk so that you can keep up with what's going on. It's going to move fairly quickly, hopefully little bits and pieces about different research projects. So the first thing I want to talk about just generally is, how did I become a neuroethologist? Like, how do you do this? How do you end up studying the nervous system of sea slugs, right? It always boggles my mind that this is what I do. And I always start off with this whenever I give a talk. And only one time did I have somebody raise their hand and say, I did. So my friend, my colleague at Cal State East Bay, who worked on many of these projects with me, decided he wanted to do sea slug neurobiology when he was 10. I don't really understand that, because I didn't even know I wanted to do anything other than football until I was in college. So it was very weird. But I actually didn't even start off in biology as a neuroethologist. I started off as a sea turtle ecologist. And so I want to talk about how I developed developed an interest in understanding the neural aspects of behavior by talking about the first project I did as a graduate student, looking at how sea turtles do natal homing. The next thing I want to do, or I'm going to do, is talk a little bit about the techniques that we use in neuroscience. Not all of them, but a few of them that you'll see when I talk about the actual more hardcore neurobiology that I've been doing. Next, I want to talk a little bit about the experiments we've been doing with Tritonia diamedia. It's a sea slug to understand orientation behavior, and specifically magnetic orientation behavior. And finally, I'm going to talk about an experiment that Jim Murray, who's at Cal State East Bay, and I did looking at what happens when you have cues that are in conflict, and what might be some of the neural mechanisms that allow animals to make decisions. Okay, so this is how I started getting interested in biology in general. I grew up in Palm Beach, Florida. I spent a lot of time when I should have been in school doing this. This is actually a picture of a summer trip to Costa Rica. We were surfing, so I spent a lot of time in the ocean. When you're in the ocean, you're out sitting on your board waiting for waves. Sea turtles would pop their head up and look at you. That usually didn't bother me. It was a little bit more disconcerting when a shark looked at you. But that's why you went with buddies, right? You dilute out the chance that you're the one that's going to get eaten. So, okay. While I was doing this, I got interested in sea turtles, and I actually spent some time working as an undergraduate, and then right after graduation working for the U.S. Fish and Wildlife Service doing sea turtle research. Okay. And what I studied were these animals. These are little hatchling sea turtles. This is a leatherback sea turtle. You can see the size. It's about, the size of my friend's palm here. The smaller leatherback sea turtles here can get up to 1,800 pounds. Okay, the first time I saw one coming up on a beach to lay eggs, I thought a Volkswagen was coming up out of the water. That's how big they are. My job there was basically to look at the nesting behavior of turtles and predict how many hatchlings were produced by a specific stretch of beach just north of Palm Beach County. It was part of a wildlife refuge, so I had 6 miles of beach that nobody else could access. And so I would go out, do my research, had an ATV to get up and down the beach, put my surfboard on the back. When I was done, got to go surfing. Okay. And so this is what I do. I'd get up around 5 in the morning, go out to the beach, drive up and down, and I'd look for these. These are tracks. This was a female turtle who came up and nested on the beach and then crawled back. Sometimes they come up, they don't like the sand, so they go right back, called false crawls. And then I would excavate the nests and predict the number of hatchlings. But obviously I didn't go surfing on this day, but certain days when I was out on my board and I'd be thinking about, well, it's really interesting. The female turtles come all the way back to the same beach they were born on. And how did they get there, right? You're sitting out on your surfboard looking around, and if I have to find the Bahamas, I would have no idea which way to go, right? This is pretty featureless terrain. So how do these turtles actually find their way back to their nests? And that's what I got interested in. And it's not just sea turtles that have these. Some of the sea turtles will actually navigate over 2,000 kilometers from their feeding grounds to their nesting beach. Other animals do similar types of amazing migratory behavior. My favorite is this bird right here, an Arctic tern. The Arctic tern weighs about 13 ounces. It nests up in the Arctic, so all these little green sites are specific nests for the Arctic terns. Then they fly all the way down to the Antarctic to overwinter, and then they fly all the way back to the same exact nest. Not the same area, the same exact. How do these birds undertake this rather large migration that's very specific? We see this in other birds, maybe not as dramatic, but here's a fox sparrow on the west coast of the United States. The green areas are breeding grounds. Animals that breed up in the Aleutians actually come all the way down and overwinter in Southern California. Makes sense, right? So hang out at the beach and eat seaweed, and then fly back up to breed. This behavior is not just limited to birds. We see this in many marine organisms as well. So, for instance, whales undertake rather long migrations, going from one specific location to another. We see this also in trout, so you might have seen these around here, right? Who fishes? I don't. But if you fish, you've probably seen these. So they go through migrations. I'm much more interested in these animals. These are invertebrates. 94% of all species are actually invertebrates. So us vertebrates are sort of this little afterthought on the evolutionary tree, just a sort of accident. These are the really important animals. Okay, and what happens, these are Caribbean spiny lobsters. They live, and this group lives in the Bahamas, and they typically do not come out during the day, and they're solitary. Okay, so they go out and they forage, they're solitary. However, when the first big storm of the winter comes, they become highly social. They come out in the middle of the day, they form these big groups of lobsters, and then they start walking offshore, head to tail, like we did when we were in kindergarten. You know, they take you, go get your picture taken, and you have to hold your the student in front of you, right? And this is exactly what they do. It's called queuing behavior, and they move right offshore into deep water where they won't get disturbed by high surf action during the winter months. So the question then is, how do animals navigate? How do they know where to go? How do they follow a specific heading? And so when we think about migration, migration requires the use of directional cues to guide animals between habitats. Now, several cues are often used either sequentially or based on the most current available cue. So what this means is many animals use a sun compass. But if the sun isn't available on overcast days, many birds switch to using a magnetic compass. So it's one of these rather complex behaviors, and it's very, very interesting to me to figure out how animals actually migrate. And so what I'm going to do is talk a little bit about hatchling sea turtles. These are loggerhead sea turtles, and some experiments we did. So one thing you have to understand about many migratory animals is that they can actually magnetic fields. Okay, so they actually have sensory mechanisms analogous to an eye that allows them to detect the magnetic field of the Earth. Interestingly, when we look at the magnetic field of the Earth, there's much more information available than just directional information. So for you, I could give you a compass needle, right? And the compass needle points north, and you can maintain a given heading. But if I were to put you in the back of my Prius, my little Prius, put a little mask over your face so you can't see anything, drive circuitously around Eastern Oregon and drop you off in the woods somewhere, and said, go back to campus, would you be able to do it? Yeah. What if I gave you a compass, would you be able to do it? Most likely not, because you'd have to know where you are relative to the campus to set the proper heading, right? So you have to have this map sense. And it turns out that there's information inherent within the Earth's magnetic field that could provide this map sense, analogous to a GPS, okay? And it turns out that we think that many different animals have this sense. And so if we look at the Earth here, this is a schematic showing an idealized Earth's magnetic field. Now, at the field lines, as they come out of the Earth, make a right angle with the surface of the Earth. Do you see that? That's called the inclination. Smaller. They do not intersect the surface of the Earth. And as you go forward, you see the same thing. A measure of latitude. That makes sense? Okay, so we have now our latitude. It turns out that there's also another component of the Earth's magnetic field that varies, known as the intensity, okay, or declination. So this is a map of the magnetic inclination, shown in black, and the field strengths in red. And location on the planet has a unique declination. Okay? So if the animals can detect these two different parameters, they could potentially have a map of the entire Earth in their brain. Okay? And so I was very interested in, do turtles actually have this magnetic map system that they can use to navigate through their environment? Okay? And just to explain this a little bit better, this is true of any type of system where you have two different variables or two different parameters that vary in a regular way in directions different than one another. So for instance, we call these bicoordinate maps. You have parameter 1 in red, and it's varying from here to here, so it's increasing in strength in this direction. Parameter 2 is increasing in strength in this direction. So they're varying, and each location has a unique combination of strengths of parameter 1 and parameter 2. This is much like our latitude-longitude system, right? Our latitude-longitude is an orthogonal map. These are non-orthogonal, okay? But they still provide relevant information. So magnetic declination and magnetic inclination both vary relative to one another. So it could provide a map system for turtles to navigate. So my major professor, Ken Lohman, and I wanted to test whether or not turtles might have this sort of system. And I want to talk a little bit about the life cycle of sea turtles so you can see why we tested what we did. Okay? And like I said before, female sea turtles come up onto the shore at their natal beach. They lay a clutch of eggs, and then they go off into the surf. About 60 days later, these eggs hatch. For loggerheads in South Florida, that's about 120 hatchlings. These little hatchlings then need to get to the surface. They can't dig, actually. They just all move around. And as they wiggle, the sand filters underneath them, and the whole clutch just rises up to the surface. They stop right at the surface until the sand cools down. Once the sand cools down, then they emerge. Can anybody imagine why that might be? When is the sand going to cool down? At night. They emerge. The predators can't see them as well. Unfortunately, even despite that, we predict we estimate that about 1 in 10,000 actually make it back to reproduce. Okay, so high mortality rates for these little turtles. The turtles have to get off the beach. They go into the water. They then do migratory circuits around the North Atlantic. I'll show you an example of this in a minute. And they spend about 20 years going around a migration throughout the North Atlantic. They then settle out into feeding. So they go same time and just form a wall offshore as the females come in, so trying to get mates, get the mating when they can. Okay, so they undergo these migrations. And it turns out that the hatchling sea turtles, what they do is they undergo incredible migrations. So we do our research down here off the beaches in Boca Raton, Florida, and we'd collect turtles. But the turtles that hatch there actually swim off into something called the Florida currents. Okay, so it's not really the Gulf Stream here. It's actually the Florida current. It turns into the Gulf Stream up here. They come up at about the Cape Hatteras, North Carolina. They start going east across the ocean to the Azores, down past the Canary Islands. This is actually a current system, and this current. Now the problem is they're what we call ectotherms. Okay, their body temperature stays the same as their surroundings, and so these little hatchlings get into this nice, comfortable, warm water. But there are certain locations in their journey where if they get too far north, so right here off Cape Hatteras, if they get too far north, get up near Cape Cod, they freeze and die, because we call it getting cold stunned. Over here, they get into something called the North Atlantic Drift. If they're too far north, they go up, and again, they'll end up on the beaches of England and Ireland, where they're often found dead. Okay, and down here in the bottom portion, the water's nice and warm down here, right? But if they go too far south, they get trapped in the South Atlantic gyre where there's no other loggerhead turtles. So in essence, they're lost from the population. And so what we decided to do is test, can little hatchling turtles identify where they are dependent upon the local attributes of the magnetic field at 3 different locations. Okay? So how did we do this? What we did is in Boca Raton, we built a coil system. So if you know your physics, if you have current in the direction of your right hand, so if the current's moving up in the direction of the thumb, the magnetic field lines wrap around the fingers. So we built this coil system so that we could alter the magnetic field around an orientation arena, and we could type into our computer any combination of intensity and inclination and expose the turtles within the arena these magnetic fields. You can also, little turtles are tethered to a little lever arm. As they swim, they move around and it tracks their heading, and the tether's short enough, these turtles will swim for days and days as long as their front flippers don't touch anything. Just swim along perfectly happy, and we record their behavior with different field parameters. Okay. Oh, and I just want to show you how we do this. So here's our little hatchling turtles. We made little lycra bathing suits for them. We've just used little Velcro, and then you can see a monofilament line right here that's tethering it to this system. Okay. So what did we find? So these are circle plots of our data. So we took turtles right from the beach. We went out, we dug them out of their nest before they actually made it down to the water, took them back to our lab. So we'd go to the beach around 5 o'clock at night, hang out on the beach, collect some turtles, go test the individual. And each dot here represents the mean heading of a single turtle. The arrow represents the mean heading of the group. The length of the arrow is a measure of dispersion. The longer the arrow, the dispersion. Okay? So the dotted lines are the 95% confidence intervals. And what we found is that turtles exposed to the magnetic field parameters off the coast of Cape Hatteras swam in general to the southeast. What this would do is force the animals further into the gyre system and decrease their probability water up here off of New England. Over parameters near the Azores, the animals swam to the south. Again, this would hopefully decrease their probability of getting swept up into the North Atlantic and through the North Atlantic drift and down and keep them within the gyre system. In the southern location, they headed to the northwest. Again, all 3 of these would help maintain the animal within the gyre system. So this was pretty amazing when we looked at this, and we thought, wow, you know, we've actually demonstrated that they have a magnetic compass. But I wanted to do one more experiment because I wasn't convinced. And I told Ken, we can't just look at this. What if we put them in fields up here or down here? And if they have a true map sense, they should be able to orient back to the right location, yes? When we did that, they had no preferred heading, okay? So what it appears to be is that at each one of these locations, they encounter magnetic field parameters that drives a behavior that will help maintain the animals within the gyre system. And this type of navigation is called signpost navigation. You just keep going in one direction until something changes. You change your behavior, you go in a different direction. Okay? And it looks like this is what's happening with these. Now, I just have to add this because we're all interested in our students being able to understand and read material and going to primary sources, and that's important. And I put this in. So as soon as we published this paper, we had a call from a journalist who wrote for National Geographic Today, and we explained our study to him, and wrote this article on it, on our research. And what he ended up saying was that they've proven that these hatchling turtles have a magnetic map, which is actually the exact opposite of what we showed. Okay? So again, if you want an example of why your students should go to the primary literature, here's a great example of that. Okay. But it was still fun to look at your name in National Geographic. Just saying. Okay, so this really got me thinking more and more about the neural mechanisms underlying the ability of animals to detect magnetic fields. Okay. So I went back into the literature looking at what's known, and there are several different things that we don't know about neurobiology of magnetic orientation behavior. I'm listing 2 of them here. What's the mechanism of sensory transduction? So how does the animal take environmental energy within the Earth's magnetic field and convert that into electrical energy within the nervous system that allows them to obtain information about the Earth's magnetic field? The next thing we don't understand is the processing and motor responses that generate orientation behavior. Basically, this is everything in neurobiology. We know nothing about this behavior, okay? Which is great for me because now it gives me something to investigate. It is a problematic sensory system because all of the sensory systems that we use to detect external information typically have highly specialized structures that energy on a specific receptor cells, or they're on the surface because that energy doesn't penetrate the body. Magnetic field lines pass right through our body. You could have one single cell somewhere very deep in the middle of your body that detects magnetic fields. How do you find that one cell that has a nanometer long piece of magnetite in it? Okay, so it is a very hard thing to do. So sometimes I kind of feel like an idiot for doing it because it's really, really hard. But it's a lot of fun. Okay, so now what I want to do is talk about some of the things that I use to investigate those sensory and motor processes underlying behavior. Okay? And to get you thinking in the right context, as neuroethologists, this is the way we conceptualize what happens in our brains and nervous systems. Okay? You can think of this as a flow diagram of energy or information flowing through the nervous system. Okay? Sensory inputs, this is sensory cells detecting changes in our environment and energy in with our environment. Often at the level of the sensory input, we have lots of sensory processing going on. This is vitally important for the functioning of our brain because this sensory processing eliminates about 98% of the information that would get to our brain. Okay? If you try to constantly process all the information that's coming to our brain, our heads would be so big that would we survive? If our heads were this big, what would be the problem? Giving birth, yeah. both the child and the mother would be dead. Okay. So this does allow us to filter out a lot of the information. The filtered information then gets sent to the central processing integrator. That's typically our brain, our central nervous system. Within the central nervous system, we have various pathways that allow us to process information in different ways. For instance, we have one pathway that processes visual information that tells us what the color is. Another one tells us whether something's moves. Another one tells us, is this a person we know? Okay, so all of this stuff gets done in parallel. Oftentimes, we consider this a little black box. We don't understand what's going on. We know the input, we know the output, and what I'm really interested in, what's going on in here. Okay? That central integrator then processes information and provides motor output that drives effector cells. These are the cells that actually produce the behavior that is elicited by the stimulus. Okay? For humans, this is typically muscle, but there are different types of cells that can become activated, such as glands and ciliated cells. Okay? So how do we do this? Well, this is a recording. I've stuck an electrode inside a single cell inside the brain of a sea slug. Can you hear that? Those are action potentials. So this is a recording that I'm doing, and you can see this is in the feeding mechanism of the snail, and you can see the action potentials, and then this is a rhythmic behavior. I could record from spinal cord neurons in you and get the same sort of pattern if they were the ones involved in walking. Okay, so this provides rhythmic behavior. Then I applied octopamine. Octopamine's a neuromodulator that makes him think that there's something stuck in his throat. Okay, and then it increases the length of the bouts of activity, which would strengthen the contractions and cause the food particle to move through the esophagus. Okay, so this is called single unit or intracellular recording, something we do quite frequently in neurobiology to try to understand activity within individual cells. We also, in my lab, do a lot of histology, so trying to understand the underlying tissue architecture that allows for behavior. This is a histological stain of the foot of a sea slug. So if you know anything about sea slugs, they crawl around on their foot. Okay, so that big surface area is the foot. And I was looking for individual cells that are involved in controlling this layer of epithelium, and I'll show you that in a minute. The different colors tell us different types of cells are present. Okay, the orange-ish is nervous tissue, the blue is muscles, the pink are mucus and epithelial cells. So we use a lot of this in my research. Okay, we also use a special kind of microscopy called laser scanning confocal microscopy. In this case, what it does is it allows us to label tissues with markers of molecules we're interested in. So I'm often interested in different neurons. So what I can do is a labeling molecule to identify different neurons that contain different types of neurotransmitters. You've all probably heard of neurotransmitters. Serotonin is a neurotransmitter, acetylcholine, various other molecules. Okay, I won't go through all of the neurotransmitters because it would take all afternoon, but I do want to show you how we see different tissues. Now, this is part of the brain of a sea slug. This is the cerebral ganglion. This is the pleural ganglion. This controls what little thought they actually have. This controls the organs, so that's why we call it the pleural ganglia. And I've labeled this for a specific molecule called nitric oxide synthase, which shows us the cell nitric oxide. Okay, so nitric oxide is a neurotransmitter. And instead of taking sections where you have to section the tissue at about 4 micrometers, what we can do is label the entire brain, then use a confocal microscope to optically section the tissue. Okay? And so what I'm going to show you now is a movie of a series of optical sections that I took from this brain. Okay? So what you're going to see are areas of, this is a grayscale image, so where you see white is where nitric oxide resides. Okay? And so what you can see are different neurons, the axons coming out, neurons on the other side of the brain. Okay? This allows us, again, to optically section this. What's great about this is it gives us a spatial distribution very readily. The other thing we can then do is take that information and generate 3D movies of it. So this is the same brain showing the distribution of these cells. Now I can go in with electrodes and record from those cells to see what they're actually doing in the animal. I'm often interested in the structures that are being controlled by the nervous system. So this is an example of this. This is the head of the sea slug that I work on, Tritonia diamedia. This particular appendage right here, it's great. Nobody knew that it was special. My friend and I were looking at it. This is a new sensory organ, so I got to name it. So I called it Murray's organ. So that's my friend Jim Murray. He wasn't too happy about that. But it was really fun to type in Murray's organ and send it off to the publisher. Okay. If we look really closely here, you want to see the fine detail of what's going on. This is a scanning electron micrograph. So this is a very high resolution. On these structures are little knobs with sensory hairs sticking up all over the place. Okay? Turns out that these are olfactory, so they're detecting odors in their environment. And I'll be talking a little bit more about this. But scanning electron microscopy allows us to look at surfaces in very fine detail, so we can understand the mechanical properties of the tissue and how that relates to the nervous control of the tissue. Another technique I typically use is something called transmission electron microscopy. Transmission electron microscopy, instead of looking at surfaces in fine detail, we're looking at the inside of the cells. So what we basically do is embed tissue in plastic, section it at about 60 nanometers thick. So 60 nanometers is a nanometer is a billionth of a meter. These are incredibly thin sections. We have to use diamond knives to do this. And what you can see here, this is part of the brain. These are all individual synapses. These are cells that are talking to one another at the level of at the real molecular level. So each one of these little structures is a synaptic vesicle. They're about 50 nanometers across, and they're packed full of neurotransmitters. So now I'm going to talk about some neurons in Tritonia and how I got into actually studying mollusks, right? I started with sea turtles. How do you end up with studying sea slugs? Well, I can tell you the first thing is I wanted to get into the brain. You're not going to get into the brain of a sea turtle. So the adult sea turtle's 350 pounds if it's a loggerhead. The reason they're called loggerhead is they have necks. They're big necks because they have lots of muscles, and they'll bite right through a 2x4. Okay? I don't want to mess with the sea turtles. The second thing is if I cut the skull cap off of a turtle, put electrodes in there, and then took that to a turtle meeting and presented it, I'd probably get shot, right? Okay? So instead, I wanted to find a good model for understanding the nervous system. And it turns out that mollusks are amazing models for understanding the nervous control of behavior. And in fact, the first animal in which we understood how individual neurons worked was the squid. So the squid has a giant axon. In 1950s, they were actually able to understand the changes in permeability of the membranes that allow the nervous system to function. Beyond that, the first animal in which the neural mechanisms of learning and memory were first investigated wasn't a sea slug, but it was a related organism called Aplysia californica, also known as a sea hare. So it's very much like a sea slug, except for it has a little tiny, they're about this big, but they have a little tiny shell, residual shell, hidden under its mantle. So they're not true sea slugs because they actually have a little bit of shell left. But mollusks have been very important in our understanding of how the nervous system works. So why did I pick Tritonia? Well, Tritonia has a relatively simple nervous system. What I mean by this, humans have, hopefully, about 100 billion neurons in our central nervous system. Okay? Tritonia has about 7,000. Okay? So many fewer neurons to contend with. Often these neurons are large, and we can individually identify certain neurons from one animal to another. For instance, entire PhD was done on the left pedal 5 neuron. So I could go from one slug to another, look at that specific neuron, and figure out what its job was. And in addition, they will respond reliably when partially dissected. So I'll show you an example of this. And in fact, you can take the brain out of the animal, put it in a dish. It'll stay alive for 5-6 days, and it generates normal patterns of behavior. We call it fictive behavior. You can actually stimulate one nerve, and it'll make the brain think it needs to swim, and it'll play out that pathway. So very powerful organisms for studying the nervous system. Okay, so what is Tritonia? So Tritonia is a sea slug, lives in the bottom of the Puget Sound. You can actually find them up into British Columbia and down into Northern California in protected marine environments. They live on sandy bottoms. They eat soft corals called sea pens. The sea pens can grow about this big, and they grow in big fields. They're like wheat fields of corals. Okay. They are heavily chemically defended. What I mean by that is their tissues have nasty metabolites, so when animals eat them, they spit them right back out, except for one animal, Pycnopodia helianthoides. Okay, this is called the sea star, the sunflower sea star. It can be about a meter from one arm tip to the next. Okay, these are big animals. And so Tritonia has the ability to detect and swim away from these. I don't know why it didn't start, so hopefully it'll run. No. Uh-oh. We had to switch over the computer logged into something else, and it's not going to run. I hope some of the other videos will run. We'll see. Okay, so basically what happens is this animal swims away. It basically flattens its body and does a series of dorsal ventral flexions. That pulls it up into the water column, and the current carries it away from the predator. It's a really cool video. Okay. Yeah. So it can't sense it at a distance. As soon as the predator touches it, it goes through this behavior, and it's really interesting behavior. What to me though is more interesting is, you remember this diagram showing the flow of information? We can actually map that onto a circuit of the swim, the set of cells involved in the generation of swim in sea turtles. in these sea slugs. So there's a set of cells called the S cells. These are the sensory cells that detect the touch of the Pycnopodia. They synapse onto TR1, which is the trigger neuron. That synapses onto the DRI. In fact, if you activate the trigger neuron, you can generate a swim behavior. DRI is called the dorsal ramp interneuron, and it activates a set of cells that generate rhythmic output like you saw previously. I'm not going to go into much more detail, but it just shows you that we can map out and create models of these neural pathways and connections. And this is what we're hoping to be able to do ultimately with magnetic orientation behavior. Okay. The other thing, and the other reason we used Tritonia, and I got interested in this, is that they actually do respond to magnetic fields. So my PhD advisor put a bunch of sea slugs into an orientation arena, and they all headed in the same direction, depending upon the time of month. We could then use that information and drive them into either arm of a Y maze using the magnetic field. Okay, so we're able to show that these animals detect magnetic fields. And they have this wonderful brain. So this is the brain of a sea slug. I've done nothing except for cut it out of the animal, so no staining, no dyes or anything. Each one of these is an individual neuron. This neuron up here is the metacerebral giant. It can be 1 millimeter in diameter, basically the size of a fruit fly. Okay? If you stick an electrode in this and drive that cell, the animal starts to eat. It's the master regulator of feeding. These 3 cells, pedal 5, pedal 6, and pedal 7, are the cells that I typically work on. And the reason I work on these cells is that they're known to be sensitive to the magnetic field. Okay? So when you rotate the magnetic field around these animals, those neurons tend to fire. So how do I do this? This is a semi-intact preparation that I use in my laboratory. Here is the sea slug. I make a small incision in the back and exposes the brain. I put the brain onto a little platform. I hang the animal in the water, and I can put electrodes into various cells. That's what these are. They record the activity of individual cells. This only has 2 electrodes. I can get up to 8 electrodes to record from 8 different neurons at the same time. You might say, well, that's kind of mean. But these animals seem to be content. They'll feed, they'll mate, they'll crawl, they'll swim, they'll do all the things they normally do with their little brain on a platform. Okay. Here's actually a view microscope of this animal, and what's really nice for me is that not only are the cells that I work on large, they're also white, so they're really easy to find. So I can easily go back and look at those same cells. I also build a magnetic coil system around my recording. This is called, we call these rigs as neuroscientists. So here's my recording rig. There's a bunch of electronics over here allowing me to record from these cells, and I can rotate the magnetic field around the animal while it's stationary within the aquarium. Okay. Now, the cells that I'm interested in are these cells right here. So this is the pedal 5 neuron. And one of the ways to study individual neurons is to understand what type of neurotransmitter they use. This is a series of 15 different confocal images. So those images I showed you before, you can take the whole thing and compress them into a single image. I took 15 of those stacks of 250 individual sections, stitched them together to come up with this image of the brain. This brain is about 5 millimeters across. And you can see the individual neurons. And this is labeled for a neurotransmitter called TPEP. And so knowing that it used TPEP, I was interested in what's the function of TPEP. Would that tell us something about the role of these cells during magnetic orientation behavior? Okay. So I needed to know everything I could about these cells to try to figure out their location within the circuitry controlling migration. And so the first thing I did was injected the pedal 5 neuron with a dye that allowed me to visualize the cell itself. So this is the cell. This is where there are input regions onto the cell, and these are axons that go out to the target tissue. I wanted to know the target tissue, so I then traced these all the way out, and it turns out that the pedal nerve 2 and pedal nerve 3, the ones that contain axons of this pedal 5 neuron, innervate the foot, what we call the ipsilateral foot, so the foot on the same side of the brain. Okay? So we have innervation patterns here. Oops, I'm going the wrong way. That's what it is. And I wanted to look a little bit more specifically. Okay, so what's happening here? We have our pedal peptide here. This is the TPEP. At the electron microscope level, I wanted to make sure that these neurotransmitters are localized to synaptic vesicles. So I treated this with antibodies that have little 5 nanometer gold particles attached to them, so I could identify where the TPEP is, and you can see them in synaptic vesicles here. Here's the foot of the animal, and I'll show you a picture of the foot in a minute. But this is showing where the transmitter, these are epithelial cells up here with little cilia on the top. So it innervates a ciliated epithelium. This is showing a synapse of a motor neuron onto a individual ciliated cell. Okay. So what could these neurotransmitter be doing? Well, this is what the bottom of the foot looks like. Okay, this is cilia. These are the same types of cilia that line our lungs and line the ventricles in our brain. Okay, so same sort of structures. And it turns out that the TPEPs that we isolated from the pedal 5 neurons go out and innervate the foot. Okay, if you apply TPEP to the ciliated surface itself, the cilia start to beat. Okay. And finally, spiking in the pedal 5 cells in a semi-intact preparation correlates with increases in ciliary beating. So it looks like these cells are important in controlling the ciliated foot epithelium. Now, this is an example of the ciliated foot epithelium. This is light microscopy showing the ciliated cells. I was really interested in what else was going on. This is a light micrograph. These are electron micrographs. But what I also noticed were these structures. This is a mucus goblet being secreted from the foot epithelium. These are mucus glands underneath the epithelium. And you can see in this series of electron micrographs, all of these little circular things are different types of mucus. Okay, so there's at least 5 different types of mucus and different types of mucus-secreting cells out in the periphery. Now, this is very important for animals that use cilia to crawl. Tritonia. Tritonia is almost neutrally buoyant, so it crawls over the surface by beating cilia. To have a nice viscous environment where ciliary beating can be very effective, it needs to be, it secretes mucus. Okay. And also, mucus seems to have other roles as well, but I'm not going to talk about that right now. And in fact, with the scanning electron micrograph, I stimulated the cells, froze the tissues, looked at it in the scanning electron micrograph, and you can see mucus goblets being secreted from the surface of the foot. Okay. And in fact, if we stain specifically for mucins, and that's what this is, we can see these huge mucus glands. They come down, and you can see them how they go right through the epithelium, where they can secrete the mucus on the surface of the foot. The other thing I want to point out is, do you see all these little pink structures right here? These are muscles, and they wrap around the mucus gland. Okay, that's going to be important in a second. Okay. So the other thing I wanted to know is, okay, it controls the ciliated foot epithelium, but there are other neurotransmitters, and it turns out there are. So these cells also produce acetylcholine. Okay, and it produces acetylcholine, and the axons go out to the foot, and this is the foot here, and these are the ciliated cells, and let's see if this will work. Okay, so here's a video, and do you see these circular structures of acetylcholine? They're wrapped around those individual glands, okay? And what we think is happening is that when the pedal 5 cell's activated, it's driving the ciliated cells, making the ciliated cells beat, and at the same time causing secretion of the mucus. And it's really cool. I'm not going to show you videos of this because I'm pretty much out of time. But what happens is those muscles around those glands get activated at the same time the cilia gets activated, and it causes secretion of the mucus, much like a zit. It really looks like somebody popping a zit when you see it. It just comes rushing out. And so what we've done is done sections of live tissue, applied acetylcholine, and measured the rate of mucus secretion, and we see that it correlates very tightly with the increased activity of the ciliated cells. Okay, I think I'm just going to end there. I've got some other cool videos, but I doubt they're going to work. And so what we're doing in my lab now is following up on this by looking at the nervous control of mucus secretion and cilia locomotion in various different types of mollusks, including Limnaea stagnalis, which is a pond snail, and we have some students here, Beth and Caitlin, that are working on that. And I'm also now started a collaboration with Julia Sigwart from Berkeley to look at the evolution of nervous systems looking at chitons, which are intertidal marine organisms. If you ever want to see one, just come by my office. I have an aquarium in there with 55 chitons, although I think maybe half of them are dead. But you can still come by and look at it. So, and that's all I have. So.
Does anyone have questions?
We just use this mic here.
Teresa? Okay, that work.
So you mentioned at the very beginning, when you were describing for people like us, lay people, what kind of being in touch with the GPS system or something like this, right? And you said that our currents have the same kind of mechanism, and they always go back to the nest that they were born. So what happens when the nest isn't there anymore?
So actually, they don't go back to the nest they were born in. The terns, they go back to their own nest. So they make it, and they go back to their own nest. The fledglings actually meet somebody else, and they go off on their own, and they create their own nest. But then they'll go back to their own nest. So it turns. If that disappears, I'll make a new one. I mean, they have to spend a heck of a lot of energy to build a new nest, but that's about it. Yeah. And exactly. But we don't know in birds yet whether they actually have that sort of system. And in fact, the hatchling sea turtles don't, but Ken Lohman's lab has now shown that the adult sea turtles do. And so it seems to be part of learning as well. So as they go around the gyre as adults, they seem to get information about the magnetic field. The thing you have to remember about my research is those little hatchlings had never been in the water, right? So this is a innate program that's being played out inside their brain. As they go around, they alter that based on experience. So, yeah. Sure. Nancy.
Can you talk a little bit about the magnetic aspect of what you just looked?
Yeah, so the magnetic aspect, what I'm really interested in Tritonia, and I didn't get enough time to talk about a second experiment, is looking at what are they using the magnetic field for. And they seem to be using it because they're slugs. They do everything slow, okay? And there's a reason we call people, you say you're a slug. It's not because you shrink up in seawater either. It's because you're really slow. So basically what happens is as they get a scent of something, they have no eyes. They have 5 photoreceptors on either side of their body, so they can tell light from dark. Basically, they use odors. Odors are being carried by the currents. And so they smell an odor, and they say, I want to go in that direction. So what they do is use something called rheotaxis. So they start going upstream. But they're so slow, by the time they get to their destination, typically the currents have changed. And so it typically takes them 15 minutes to get to their goal, whereas the currents, we've done measurements, change on the order of about 5 minutes. And so what we think is happening is they maintain a heading based on the current for 5 minutes, then they switch over and they've now entrained the heading and they continue following the magnetic field and ignore the currents. And we've done data, we've collected data on the appendages that allow them, that Murray's organ, those little sniffing organ, and they shut that off. So after about 5 minutes, they have neurons that go out and just say to the sensory cells, stop working. And the sensory cells stop working, so they aren't sniffing anymore, and the only thing they're following is magnetic fields. So is that sort of what you're asking? Yeah. So it's really interesting what they're doing. And I really, really want to find the sensory cells, but I've worked on this because it's something that's tractable, and this is very amenable to research. Finding those cells, it could be one cell in the entire body, and so I still do that work, but one of my PhD advisors gave me some really good advice. And when I was doing my PhD and he knew what I was trying to do, which was find those receptors, he told me that the value of your dissertation is equal to the impact on the scientific community divided by the time it takes to complete that work. Okay? And it really told me I better find something that I can get done with, and just in case we can't find the magnetoreceptor. And so we've got that going on in the background. It's finding the right techniques and so forth. But what's really interesting about the work that I'm doing now on the ciliated cells is that the neurotransmitters that the molluscs use to control the cilia are the same neurotransmitters that our body uses to control mucus secretion and ciliary beating in our lungs. So it has direct consequences for understanding cystic fibrosis, hydroencephaly, and many other ciliary dysfunction diseases. So, yeah.
Any other questions?
Yeah.
So where this is going on with, the brain in a human being or a more complicated organism, would this even reach the level of being in the brain, or would this be some subsystem closer to where the brain is?
In terms of...
Oh,
right.
Right. I wonder if that signal would really travel to your brain, or whether there's just a local...
Right, and so there probably are local control, but that information always gets sent to the brain because your brain's coordinating everything that happens in the body. So even our reflexes, that you'll see a picture and a diagram in the textbook showing you your patellar reflex, and it leaves out the fact that information still goes to the brain. Even though it acts independently. And in fact, we have a third nervous system in our body called the enteric nervous system, which controls our gut. It's independent of our brain and the rest of the nervous system, but it still sends information to the brain so that the brain can regulate the processes that go on. So, yeah. So everything goes to the brain. It might not reach the level of consciousness, right? But everything goes to the brain. So, yeah.
So, thinking about the other question, the original question of locating the mechanism of the sensory organ for magnetic sensory, what kind of experiments are you setting up to try to zero in on that?
So there are two different competing hypotheses for how animals detect magnetic fields. One of them is that there are cells within the eye, especially of vertebrates, that have molecules called cryptochromes that I'm going to say this very specifically, are thought to potentially be influenced by the magnetic field. There are similar molecules that have been shown to the actual chemical reactivity can be dependent upon the magnetic field, but not of the cryptochromes. So that's one hypothesis. But the animals that I work on have no eyes. And in fact, I do some work with spiny lobsters. I haven't talked about that. We actually cut the eyes off. If we didn't cut the eyes off, somebody's going to throw them in a boiling pot of water. So, so they don't appear to have a similar mechanism, but they do possess magnetite. So they have little pieces of magnetite, but the magnetite's really small. And so what we've done with Tritonia and with the lobster is grind them up, and we've been able to extract these little tiny 100 nanometer length particles of magnetite. Now it's trying to find them. So I've been sectioning, I sectioned an entire spiny lobster, stained it for looking for magnetite, but now I've got 6,000 sections to look at. So it's not an easy thing to do. So we sort of have it going in the background. What's really probably going to help is, if you've heard of CRISPR, this is a new way of dealing with different cells, and we're hoping to be able to use that system, and it gets efficient enough across different species to start to look for the individual cells that are active. The cells that I'm working on here, unfortunately, it's hard to work back up in this system. So going from the motor neurons up into the brain out to the sensory system, there's no good way to do that, except for just sticking electrodes into all the different neurons. So, yeah. Well, thanks everybody for coming. I hope you learned a little bit. Sorry about the one movie. I'll post it on a website, and if you want to look at them do their swim behavior, you should. It's really, really cool. to watch these animals swim. Although, let me tell you, if you do watch it, realize that we take a very liberal interpretation of what swim means, okay?